Hello, everyone, and welcome to today's webinar, Using Metabolic Analysis to Optimize Bioprocess Conditions. I am Matt Atchett of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Biolog. To learn more, visit biolog.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Stacy Montgomery, Senior Director of Marketing at Biolog. Stacy, you may now begin your presentation.
Thank you, Matt, and thanks to everyone joining us for this webinar on how Phenotype MicroArray technology is used to optimize bioprocessing. Two important aspects of optimization are the development, selection, and characterization of production strains, and how to develop optimal growth conditions to produce the highest yield possible. In the next few slides, I'll show you how the technology works to provide a comprehensive global picture of your production cell lines and how you can use this technology in different stages of development. There are many ways to take a comprehensive or global look at cellular properties. The issue is that these transcriptomic or proteomic approaches often show many changes at the transcription or translation level, but there may be downstream modifications or processes that result in no discernible effect on the cell. So this means there's an enormous amount of data to sift through to find what is actually meaningful and actionable. So while they result in massive amounts of data, it can be difficult to determine how those changes actually affect a cell. So we've taken a different approach and instead focused on cellular phenotyping using a universal respiration reporter that we can link to many different compounds or conditions to get a functional metabolic readout. So we have a series of redox reporter dyes that allow us to link cellular respiration to our compounds of interest or different conditions. If the organism can utilize the compound or respire in the presence of components in the well, they respire, they produce NADH, and that NADH can then be used to convert our colorless tetrazolium dye into a visible purple color. So by looking at expressed cellular properties or phenotypes, we're eliminating much of the noise and can focus on properties that are biologically relevant to the cells of interest. The platform for all our assays is a standard 96 well microtiter plate. Each well contains a different substrate or an inhibitor along with our universal respiration reporter, and if the organisms can respire under this condition, the well turns a visible purple color that's non-reversible under physiological conditions. For bacterial cells, we have a series of 20 plates. The top 10 plates are focused on metabolic conditions, where we look at carbon metabolism, nitrogen metabolism, phosphorus, sulfur, osmotic and ionic effects, pH effects, and then the second set of plates at the bottom are chemical sensitivity assays. The easiest way to think about what we're doing is that each well is testing for the presence or absence of a known biochemical pathway. These are not random chemicals that we used because they were inexpensive, stable, easy to source. They were all designed to interrogate a specific biochemical pathway known to be present in bacterial cells or a particular condition such as optimal pH, ability to withstand salt, etc. The same thing is true for the inhibitor panels. We have a wide range of inhibitors with different mechanisms of action. So it's like we're poking the cell in different places to see how it responds to all those different insults. These assays are very simple to run and consist of the Phenotype MicroArray plates, the OmniLog incubator reader, and software that's designed to interpret the results. So this is an example of a typical experiment where we're inoculating 2 plates with different cell types. So you can think of it as a wild type versus a mutant strain, or it could be untreated cells versus cells treated with a particular compound we're interested in. But we inoculate our plates, one type labeled in red, another type labeled in green, place it into the OmniLog system. It takes a time point every 15 minutes, and then we get the plates with the peaks showing the phenotypic activity. The software then allows you to overlay those particular curves for the different cell types, and everywhere you see yellow, the cells are the same. If you see a red peak or a green peak, it means there's been a change in phenotype between the 2 strains that you're comparing. As I mentioned, these are kinetic curves for each well we're looking at, and we're looking at respiration over time. And then we can investigate the lag, the slope, or the maximum response for each substrate or test condition. This technology can be used for 2 important aspects of bioprocess development: strain development and selection of clones, as well as optimization of culture conditions for best yield. And we'll look at those 2 aspects of the technology. The typical methods for process development are slow and inefficient. So on the left side of the screen, we typically start with a production strain, modify that strain, creating daughter cell lines with better production traits. We then choose the clones that we're interested in and manipulate the growth conditions such as the media, the pH, different conditions, and then we screen a large number of those clones for increased yield and titer. So this is this iterative circle where we start with the strain, mutagenize it, select clones, measure their titer, then we grow under multiple feed conditions, measure the titer again, and if you get lucky, we proceed to scale up. If not lucky, we start over. So this is a process that oftentimes gets repeated over and over again. So let's review a few different kinds of experiments that can be used to analyze or compare your strains. This first one is a typical phenotype microarray comparison of 2 strains. It's a wild type MG1655 E. coli strain and a mutant that we've developed by transposon mutagenesis. In this case, we've inserted TN10, which carries a tetracycline resistance gene, and we've inserted that into the MALF gene. And what we see when we compare the wild type to the mutant in this experiment is we see that we've lost the ability to utilize maltose sugars as we expected. And we can also see that we gained resistance to tetracycline and confirm our genetic construct. Another thing we can see by looking at this is we can see that we didn't have any off-target effects. This is a more interesting example of mutating a regulatory gene. In this case, we've interrupted the OxyR gene by inserting a kanamycin-resistant cassette. So the OxyR gene that we've mutated here is a global transcriptional regulator of the oxidative stress response. And we can see that this mutation affects a variety of cellular phenotypes. And this is a very interesting example of being able to globally or comprehensively screen your cells. If you're dealing with a regulatory gene, sometimes it's hard to predict what other genes this is affecting. So in this way, we can see that we've mutated our gene, our OxyR gene. We can see that we've introduced aminoglycoside resistance, and we can see a whole range of changes as a result of this global transcriptional regulator. Sometimes there's a need to discern the differences between 2 closely related strains. In this case, we're comparing 2 strains of the same species, a pathogenic E. coli 0157 as compared to MG1655. And people have understood that 0157 behaves differently from typical lab strains of E. coli for a long time. In this example, everywhere you see a black box, the software is flagging a significant difference between these 2 strains. So experiments like this can be really useful to determine changes in strains due to phenotypic drift. For example, if something feels off about your production strain, you can use these types of experiments to detect what those changes might be. And then later, you can also use this as a very comprehensive QC test to ensure the strain that you're working with today that you'll be using behaves exactly like the strain that you were using 2 years ago. So how do we correlate these color changes in wells to the metabolic function and figure out exactly what's going on with our strains? So let's take a look at the OmniLog software and see what the options are. So the OmniLog PM software is linked to the Kyoto Encyclopedia of Genes and Genomes, or the KEGG database. And so we can click on a single well on a plate, and the software will tell us what the compound is in the well, along with the mechanism of action. In this case, we can see I've clicked on a well A3 in PM plate 1, and I can see that this is N-acetyl D-glucosamine, and it's a carbon source. One more click, and it will show me the molecular structure of that compound. Another click, and I can get to the enzymatic conversion and the products of that conversion. Another click and we can get to the enzyme that catalyzes the conversion. And another click, we've got the amino acid sequence and the nucleotide sequence of the gene for that enzyme. So within moments, we can get to this end result, which is a pathway analysis at our fingertips. So to summarize, we looked at the technology and how it can help us develop, select, and characterize clones with useful properties. Now we can look at how it can also be used to determine optimal conditions for growth of our cell line. So once we've selected our specific clones, it's time to optimize growth conditions. We can now take our prospective cell lines and determine the optimal carbon source, nitrogen source, pH range just by inoculating the proper plates and incubating overnight. This is much more efficient than performing shake flask studies for each individual compound or condition on multiple clones. So in this way, in an overnight experiment, we could actually test 1,920 conditions for each of the individual clones. The goal is to find optimal culture conditions as early as possible. And this is some work that was shared by Nigel Mounsey when he was at Dow AgriScience. And his goal was to determine optimal conditions for the development of a new product. So he was looking for carbon sources that supported growth. And in addition to measuring carbon source utilization, he also tested the titer of the product in each well. Those wells that demonstrated a decrease in titer were labeled in green, and those that had an increased titer were labeled as purple. And what he found was culture conditions that support the best growth don't always result in the highest yield of product. So we need to find the optimal carbon source and conditions for growth, but also for optimal yield. In addition, while we're doing this, we can look at alternative carbon sources that may be less expensive, and once we get to scale up, this could end up saving us millions of dollars in production costs. Now we'll take a look at an experiment where a client wanted us to examine their production strain along with several daughter cell lines and provide input on optimal growth conditions. So we ran their parental strain in our lab and 3 of their daughter cell lines on the PM, or Phenotype MicroArray, metabolic panels 1 through 10. So when we tested the parental strain and looked at the baseline metabolic response, what we saw was a little bit surprising because it was pretty non-reactive. If you look at the PM panels labeled 3, 4, 5, 6, 7, and 8, you can see that there weren't a lot of positive reactions. What we found out upon reviewing the data was that the client's production strain actually required leucine for growth. This need was unknown to the client, but made sense based on the formulation of media that had been shown to be reactive with this strain. So they were dealing with a leucine auxotroph. The client wanted us to continue testing the daughter cell lines to find out more metabolic information if we could. So we found that all the daughter cell lines, when we compared them, showed increased sensitivity to salt. So we exported the data and compared it side by side in a graphical format. And you can see easily that the daughter cell lines had become more sensitive to salt than the parental strain. So our observations for the client were that the parental strain and daughter cell lines were all leucine auxotrophs, and all three daughter cell lines were more sensitive to sodium chloride concentrations. So they need to include leucine in the production media and manage their salt concentrations. The examples I've shown you so far have been in bacterial production strains, but the technology also works for yeast and filamentous fungi. And this is an example of how a single microplate was used to look at carbon source utilization, secondary metabolite production, antimicrobial properties, and they actually used it to dereplicate their culture collection. So this is work that was done by Maya Singh at Wyeth a number of years ago using a single FF microplate that was originally designed to identify fungi to the genus and species level. What he did with the plate, he took his culture, they don't tell us what it is in the paper, they don't tell us what they're trying to produce, but you can guess from these experiments that he's looking for production of a secondary metabolite that inhibits Candida albicans. So what he did was take his strain of interest, inoculate the FF microplate and incubate it. Then he read it on a standard plate reader to see which carbon sources in that plate supported growth of his organism. For those substrates that supported growth, he then scaled up into 1 liter shake flasks and measured the target metabolite. He also took the supernatants from the FF microplate and then used those to test against a lawn of Candida albicans to see if there was antimicrobial activity in the supernatant. What he found was that gentabios and D-fructose supported the highest levels of growth. But on his scatter plot where he was measuring the compound of interest in the shake flasks with the substrates that were best for growth, the highest result was obtained with sucrose and L-rhamnose, not fructose or gentabios. And when he tested those supernatants from the FF microplate for antimicrobial activity with Candida albicans, sucrose and rhamnose were the substrates that led to the highest inhibition. So we can see from these experiments that the preferred carbon source can be decoupled from the carbon source that will allow for the highest yield or titer of our product of interest. So I've shown you how the technology can be used for all types of microbial cells for clone selection, characterization, and determining optimal growth conditions. I also want to mention that we have the same technology optimized for mammalian cells as well. So it's the same basic technology, but the compounds and protocols were optimized for interrogating mammalian cell lines. I've covered a couple of use cases on strain development and characterization, as well as optimization of culture conditions leading to higher yields, but we've also seen our technology used in novel ways for a lot of other applications. So I've placed a link to a publication database on this slide, so I would encourage you to go to this link and look at the publications. You can sort them by organism type, application, product type, so it's a great resource that can also be sorted based on countries or location of the authors. So it's a great resource for you to use. And with that, I'll take questions.
Yes. Thank you, Stacy, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Okay, let's get started. Our first question today. Number 1, can you use this technology with other reporters such as GFP?
Yes, you can. So our OmniLog instrument can't read fluorescence or chemiluminescence, but the plates are set up such that you can add dye if you want it, put it in the OmniLog and read the purple color formation. So do the metabolic respiration. You could also have, if there was a green fluorescence protein or another reporter, you could read those on another plate reader. So they will work together.
Okay, thank you. Next question. Is it possible to order custom plates where we can choose only the compounds we're interested in?
That's a good question. So we have done some special pilot projects for specific customers, but at this point in time, it's really a process where a person would have to order at least 100 plates of that type, maybe even more, before we could take a project on like that. So I would say it may be possible. The best thing to do is to send an email to info@biolog.com, and a representative will get back with you and could discuss whether it's possible.
Thank you, Stacy. Next question. How can I find out what compounds are present in each plate?
Okay, if you go to the website and look under the products tab and you go to Phenotype MicroArrays microbial, you'll see that there are buttons you can press for the Phenotype MicroArray plate maps 1 through 10. Then there's another button for Phenotype MicroArray plate maps 11 through 20. Then there's a 20 through 25, 21 through 25 for yeast and fungal inhibitor plates. Same thing if you want to look at mammalian assays, there are plate maps for those too.
Okay. Question number 4. We just got, how many plates can the OmniLog hold?
So it can hold 50 microplates for an experiment. So you'll end up with an enormous amount of data in a single overnight experiment.
Thank you. Move on to the next question here. Would this work with microbiomes or mixed communities rather than pure cultures?
Yes, absolutely. So it's very interesting. There are a lot of people that are currently working with microbiomes and looking at the functional analysis of the community rather than the genetic analysis. So I would really, for that, if you're interested in microbiome analysis, I would encourage you to go to the website, to the publication database, and search microbiome, and it will pull up all the recent papers of what people have done with these plates.
Okay, a couple questions here left. The next one, have you seen any examples where the dye itself interfered with the growth of your organism?
It's possible that the dye could be toxic. We haven't tested every cell line. We've certainly tested, thousands of different cell lines with technology, but there can be situations where the dye could interfere. One place it can be problematic is working with anaerobic cells. And so then what we typically have customers do is not use the redox dye and then look at growth instead. And so they're going to read it on a separate plate reader and look for turbidity in each well.
Great. Well, looks like we have one more question for today, Stacy. The last question here. Which mammalian cell line have you applied this technology for?
We've done a lot of different cell lines. Obviously, CHO cells, colo cells, there are a number of those. So if you go to our website and just look under mammalian Phenotype MicroArrays, you'll see examples and lists of all sorts of other cell lines that people have worked on for mammalian cells.
Okay, thank you, Stacy. And before we go, do you have any final comments for our audience today?
Just thank you for joining us. We really appreciate you spending your time with us. And if you want to contact us at info@biolog.com, I think this audience is very international and we have distributors all over the world. So just contact us at info@biolog.com. We'll put you in touch with your distributor or your sales rep. If you're interested in a webinar for your group, we're happy to do that as well. So just definitely send us an email at info@biolog.com and we can discuss the specific needs that you have.
Fantastic. Well, thank you again, Stacy, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Biolog, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.
Thank you.